Okay, ladies and gentlemen, good day all. Today we are going to look at the proteins and amino acids. So let's start with actually the synthesis of protein. So generally in a cell, we have a nucleus, and in this nucleus we have DNA, and a copy of this DNA is converted into a messenger RNA, and the process of the conversion of this DNA into messenger RNA is called transcription. And then the messenger RNA will now be translocated from the nucleus to cytoplasm, where the mRNA uh, attached to the ribosomes for protein synthesis. And then another type of RNA called transfer RNA collects the specific amino acids that are needed to make the protein. And then there are over 20 different transfer RNA, and each of the transfer RNA is specific to one amino acid. And this transfer RNA will now bring amino acids to the ribosome, and now the ribosomes will now start building or assemble a chain of the amino acids in a proper sequence based on the information that is coded in the messenger RNA. And then the process of the adding and bringing the amino acids to produce a long polypeptides or a proteins is called elongation. And then the ribosomes will now continue to move down from the mRNA strand until all the appropriate amino acids are added and the protein is completed. And after this protein is completed, there is also another step, which is the post-translational modification of the proteins. Some groups like methyl group are usually added after the protein synthesis, and then the now synthesized protein will now fold into three-dimensional structure, and that is uh, the process of post-translation modification. It means that after the proteins are made, then it needs to undergo some modification before the proteins now become functional. And okay, then after we have these functional proteins, and now it will now elicit its physiological functions. So this is how all the proteins are produced in the body. A specific messenger RNA are made for specific for specific proteins, and those the information that are coded in the mRNA will now be used to build off and synthesize the proteins, and the proteins will finally be synthesized and modify into a functional one. This is how hemoglobin are produced, some neurotransmitters, some proteins that are found, all the proteins that are found in our body. This is how they are produced. And after these proteins are produced, there is also called a process of denaturation: heat, acid, salt, mechanical agitation, and also proteosomes, because some of these proteins, after they elicit their physiological functions, especially in a living system, they need to be degraded. Like for example, we have adrenaline. Adrenaline is a hormone and is a proteins, and we only need adrenaline when our body need it. And after it elicits these physiological functions, then it need to be degraded. So through the process of the central dogma of molecular biology, that is DNA to mRNA and mRNA to proteins, a lot of proteins are produced, including the adrenaline. So you see that when adrenaline are produced in our body, we usually excited. So if these proteins adrenaline is not degraded, will keep on or will remain excited. So because most of these proteins are degraded after they elicit their functions, so that is why most of our body after some physiological functions have been actually done. So they need to be digested and they need to be degraded. So also outside the physiological system, it is also so outside living system, a three-dimensional structure of proteins can be lost. Look at it, this is a normal proteins and it is in three-dimensional structure, and this three-dimensional structure of the protein can be lost through the heat, the use of acid, salt, or mechanical agitation. So that is why whenever you are working with proteins, you have to be extremely careful, because the proteins can easily get degraded, and once the protein is degraded, it means that it lost its function. So, and then the next thing, how this amino acid look like. In the amino acid, there is a central carbon, and this central carbon is called alpha carbon, or in some other sources, they are called stereogenic carbon or chiral carbon. What is chiral carbon or stereogenic carbon? Is a carbon atoms that attach to four different groups. So if you look at this, you will see that you have amino group, you have acid group, then we have side group, which is R side group, and we have free hydrogen. And each of these amino acid have its own specific the side chain. For example, now this is the aspartic acid. In the aspartic acid, you see the amino group hydrogen and carboxylic. So if you look at this, this is an amino acid. This is the structure of amino acids. So in this amino acid, we have acid group, we have amine group, and we have side chain. So, and we also have hydrogen, free hydrogen. So alpha carbon or chiral carbon, this is the alpha carbon or chiral carbon, which is the carbon atoms that usually attach to four different groups. So we call it stereogenic carbon, chiral carbon, or alpha carbon. So this side chain is what actually varies from one amino acid to another, but the amino group, the carboxylic group, and free hydrogens remain the same for all the amino acids. So in the case of this amino acid, aspartic acid, you will see that the side chain is made up of COOH. So this is the side chain here, and the side chain for the glycine is hydrogen, and then the phenylalanine, the side chain is the aromatic compound. So this is how one amino acid varies from one another. Thank you. If you don't have it, there's no problem now.